All right, guys. Today we're going to take a little break from our viruses presentation and do a little review from freshman biology. Since this is review, even though you may have forgotten it, I'm not going to necessarily make you take notes on it, and I'll go through it fairly quickly, but you can watch this screencastify as many times as you want, pause, rewind, until you remember this idea. So all cells make protein. It is their main job. And we remember that viruses are just little bundles of protein with genetic material inside, DNA or RNA. But a virus doesn't have anything to make that protein or to copy its genetic information, so it uses the cell's machinery. So we're just going to have a little quick review of the cell and going from DNA to protein. The central dogma in biology is that you go from DNA to RNA to protein. Now remember, DNA is where our genes are. RNA is a copy of one gene on that DNA. And then that RNA carries the genetic information to make a protein. So all of our genes code for a protein. Here's a review of the cell. We're not going to go through all of these things. The main ideas that I want you guys to remember is that in the middle of this animal cell, we have a nucleus. And the nucleus is where all of the DNA is stored. It does not leave the nucleus, and it can't get its message outside. So it makes a copy of RNA in the nucleus. The RNA leaves, and then it finds one of these tiny dots called ribosomes. And ribosomes are the protein factories. They're where the RNA is going to be read and translated into protein. So what organelle synthesizes protein in the cell? I just told you. So if you remember, it is the ribosome. But ribosomes can't read the DNA blueprint. And why is that? Well, first of all, the ribosomes understand RNA, the chemical messages of RNA. And DNA is too big to leave the nucleus. It must stay and be protected in that nucleus. And if you make an RNA copy, you can make several RNA copies of that gene. And all of those RNA copies can each find a different ribosome, so you can make a lot more protein that way. What base is found in DNA and not RNA? DNAs have T's and RNAs have U's. That's not really that essential for virology. So here is the process of transcription. I also have a video on the PowerSchool page. So we're going from DNA to messenger RNA. And the enzyme that does that is RNA polymerase. And you're going to remember the polymerases because viruses usually carry their own polymerases because they do this process that I'm going over and they mess it up. I'm giving you the normal way a cell does it, going from DNA to RNA to protein. RNA viruses and do it a lot differently, and we'll get into that. But you need to understand the usual way. So RNA polymerase basically comes in and makes a copy of one gene, and it goes in the 5' to 3' direction. Translation, mRNA then leaves the nucleus, and remember the DNA stays there, and the mRNA finds a ribosome, and the ribosome then assembles the protein. These little green tRNAs, transfer RNAs, bring in the amino acids, and amino acids are the building blocks of protein. So this is where our protein is being made. And remember, viruses hijack this, so the ribosomes will make viral RNA instead of the cells. Here's a little animation of that process. All right, guys, that's all I have. So review the videos, review the notes, and we'll pick up with our viral notes later on this week. All right, thank you. Bye-bye.